Hello, my name is Morgan Wood, and I am a first-year master's student at Virginia Tech under Dr. Jed Barrett within the biology department. Today, I will be describing my efforts to build species distribution models for the micro-invertebrates within the McMurdo Dry Valleys in order to make predictions about climate change, particularly weather conditions that may alter biodiversity patterns in this region of Antarctica. My co-authors on this are Mark Salvatore and Dr. Barrett. Dry valley terrestrial microinvertebrate distributions are ultimately controlled by the availability of liquid water, but soil salinity and carbon can also determine habitat suitability and community composition. Some taxa have more tolerance to wetter areas, whereas others prefer more dry and saline environments. For my research, I will be looking at nematodes, tardigrades, and rotifers. For example, the nematode Plectus has a higher tolerance to moisture, whereas Scottnema can be found in those more dry and saline soils. And we'll talk about this a little bit more later on. With the entire world experiencing climate change in some way, shape, or form, the dry valleys specifically are seeing more intense and longer melt and thaw durations, allowing for more liquid water to be present across the landscape for longer periods of time. When thinking about previous species distribution studies, scientists were able to map out current habitat suitability. However, with recent advances in remote sensing and geospatial technology, we're now able to see a bigger picture, such as viewing and analyzing liquid water across the surfaces of landscapes from space. My collaborator at Northern Arizona University, Mark Salvatore, has created water availability projections for the McMurdo Dry Valleys, projections that we have not had before. Now, how does this tie into species distribution modeling? Well, these water availability projections, along with 30 years worth of biotic data such as species abundances or binary presence absence inputs, and abiotic data such as soil chemistry, so pH, salinity, carbon, along with topography and climate data, so annual and seasonal temperature, can aid in not only creating habitat suitability models that we've seen before, but also allow for future projection of species distributions and community composition. These models will be generated using standard R packages such as Maxent or Random Forest to project future and current distribution patterns. As some taxa have more tolerance to moisture, it's imperative to think ahead. Will those taxa that thrive in dry soils like Scottnema become less abundant over time, and will other taxa such as Plectus become more widespread if melt and thaw events continue to become more intense and last longer? I also hope to analyze community composition. So, in what areas do we see more than one micro-invertebrate, or more than 2 or 3? And what makes those areas unique, or what conditions are present to form these multi-taxonomic communities? These questions and more hope to be answered by the outputs of the species distribution models, as well as other statistical analyses to look for collinearity or multi-collinearity between our abiotic and biotic inputs. The results of these models will aim to contribute to the understanding of how global change is affecting the dry valleys and build one of the first comprehensive species distribution models for the McMurdo Dry Valleys. I want to thank Eric Sokol, Meryl Mims, and Brian Brown, as well as my lab members, Meredith Snyder and Sarah Power. My funding for this project is supported by NSF through the MCM LTR grant that I am a part of. Thank you very much.